everyone. A very warm greeting to one and all. My name is Shrikari Krishnakumar, and I would like to welcome you all for today's webinar on Cheminformatics for biomedical drug discovery. This program is designed to address the challenges associated with understanding, modeling, screening, and applying cheminformatics strategies to improve drug discovery results. The process of finding a new drug against a chosen target for a particular disease usually involves high-throughput screening, wherein large libraries of chemicals are tested for their ability to modify the target. With contributions from leading researchers in academia and biotechnology, pharmaceutical industry, as well as experts from the software industry, this comprehensive mentor-guided training program explains how cheminformatics enhances drug discovery. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the omics logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Students will also have access to the omics logic resources, but how does it help advance bioinformatics teaching? Omics logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path of the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, omics logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This also comes with an access to the omics logic courses, which includes courses on bioinformatics, precision medicine, multi-omics that includes genomics, transcriptomics, metagenomics, epigenomics, data science that includes using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Users will also be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. And different stages of analysis are performed in different sections on this multi-omics platform. So now to view the various courses, you need to first sign up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, as well as a brief bio about yourself that will consist about your education background and research. And the mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the Omics Logic Learn portal before we proceed ahead with today's webinar. I am sharing the link for signing up on the portal in the chat box so that everyone can sign up. So for those participants who are new to the portal and who do not have an account on the portal yet, please click on the Create an Account option that you see here, and please enter your name, email ID, and password, and you'll be good to go with setting up the profile. Or you also have an option to sign up using any of your social media handles that includes your Google, Facebook, Apple account, GitHub account, or even Twitter. And with that, I'll pause for a couple of seconds for the participants to sign up on the portal. Once you have signed up on the portal, please put a yes in the chat box so that I know I can proceed ahead. And I'll show you the various courses and example projects that are there on the portal. The fee structure of the program and other program details will be discussed as we go along. So for now, let us sign up on the portal first. Once you have signed up, you can leave a yes in the chat box and I'll show you the various courses that are there on the portal. So we have one yes. I'm waiting for the rest of the participants as well. All right, so we do have a couple of yeses. For those participants who already have an account on the portal, you can simply click on the login now option that you see here and click on sign in. So once you sign in, you'll be able to see the various courses that are there on the portal. So you can simply enter the keyword or the course topic that you're interested to work on. So for instance, let's say you're interested in cheminformatics, you can simply enter the keyword cheminformatics and that will show up the course associated with it. So before we proceed ahead, let me briefly demonstrate how you can set up your profile. You can simply click on the tab that says welcome back with your email ID. And once you click here, you will see an icon that says profile. You can simply click on that and that will show you the various options to edit your profile image, your bio, your name, as well as link to your social media handles. And under the activity tab, the community manager and the program mentor will be tracking your learning progress. And the courses you're currently completing, as I mentioned in my slides, will appear under the courses tab. And once you complete a project and upload it on the portal, it will appear under the projects tab. Finally, when you enroll for the program, such as the Cheminformatics program, that will be displayed under the programs tab here, as well as the certificates will appear under the certificates tab. So if the participants have no further queries, let's move on to the next exciting part of today's webinar, wherein we are joined by Dr. Puneet Thakkar for an expert talk. Dr. Thakkar is a drug discovery scientist and a digital healthcare enthusiast. He has a PhD in drug discovery from Italian Institute of Technology from Genova, Italy, and with several years of experience in serving clients coming from human drug discovery, veterinary medicine, and agrochemical settings, Dr. Thakkar is a strong advocate for using advanced technology in pharmaceutical research and development. And with that brief introduction, now I'd like to pass on the stage to Dr. Puneet Thakkar, who will also be the program mentor for the Cheminformatics drug discovery. So the stage is all yours.
Thank you very much, Sanalika, and welcome to everybody. Happy New Year, first of all. And since we are beginning with this Cheminformatics size and a kind of a training program, educational program, I see a lot of opportunities in this year as well as in the upcoming years in the area of drug discovery and development. Since there's a significant penetration in the area of technology embarking into the process of life sciences and pharmaceutical. Last couple of years have been very much evident about how sooner the drug discovery should be done rather than this regular routine exercise of taking 15 years of time. We have seen the COVID penetration, how the human life got impacted, how many valuable lives have been sacrificed with the infection. And we have seen the power of vaccine, the medicine, how urgently it was required. And the impact of vaccination is also very much seen that how it could be a life saving if it is developed earlier. Not just only the COVID as a scenario, as an example, there are so many numerous unmet medical needs are in front of us where drug discovery is urgently required, where new therapeutics are urgently required, where innovative medicines are required. Without the help of technology, it would be very difficult to meet that requirement in such a shorter time. We believe the incorporation of artificial intelligence, machine learning, and advanced technologies, even the technologies which are not very much directly evident. For example, the machine learning based GPT chatbots, or a method, a technology incorporation where it can help to search a very large data set or connect the entities of pharmaceutical interest together in a meaningful way that retrieve a suitable information that a scientist is looking for a successful drug discovery can be so much beneficial. Sometimes these things are not very much clear, or which are not directly seen by the people who are into the early stage of their career. In this particular training program, we'll not only go through the traditional ways of using the technology in identification of the initial or hit molecules called hit identification, the identification of chemical compounds that can become a suitable candidate for the lead as and eventually as a drug, but also look at a different perspective, how these technology things should be seen from the angle of artificial intelligence and machine learning in every aspect, right from the beginning of searching a database. We need to see how we can automate such processes so that the larger number screening, more customizable, most customized fashion, the data can be retrieved. How you can make the workflows automated in a way that it should accelerate the process of drug discovery. Not completely end to end the automation of drug discovery, but at least some part of that process can be automated. There are several ways, several outlook to look at the same thing in different ways. We will cover those things also in the exercises that we will be doing in this educational series. We'll start with some of the basic databases, the content of the databases, and how you interpret the data in the such publicly available databases. The very good thing in those publicly available data sources is that they are freely available to serve the mankind, freely available so that you should have no boundaries, no boxing in the in the thinking that there are limitations in your mind to develop a new medicines. The data sets are high quality, they are validated, they are available to everybody. There is no restriction of how much you can download. There's no restriction of stopping anybody to use these data sets. So publicly available data sets will go through, will understand their files, will understand their processes, will understand how differently you can utilize those data sets. We'll go to the basic and simple, yet very powerful traditional methods of drug discovery utilizing those individual set of data sets. Many times we can do fancy stuff, but it's difficult to understand the underlying concept of how we are using it. Why are we using this? Is there any best way out of there? Is there any alternative advanced technology way there? Are there ways where certain methods will be replaced in the future? For example, protein modeling. The traditional ways of protein modeling have been replaced, completely replaced by the artificial intelligence method. Now, thinking someone doing PhD work in molecular modeling would be very rare, I would say. Very rare, or maybe requiring a very, very complex study that is required to be done in a PhD work. But most of the times the structures are either available, the key structures, or if they are not available, they are available by means of AlphaFold or 3D structure prediction predicted by the artificial intelligence method. We'll go through some of the complex methods and technologies, for example, molecular docking based screening, chemical similarity search screening, right? Um, so these screening methods are very simple but very powerful. Now you need to see the same data set can be done with the help of machine learning and artificial intelligence, where you can apply what are the opportunities, what are the challenges. Every technology, every method developed in the past or currently, or they may be developing in the future, they will always have the limitations and opportunities. So we need to always balance out the things, when to use which method, what is the ideal way to do that, what is more demanding in the global scenarios in terms of career objectives, future career ambitions, and what are the areas where the employability is high. That also we will look into some of the details. So overall, this training program, this educational series is packed with full of learning, mixing the traditional as well as advanced technology in place with the outlook of consulting, saying what to use, when to use, and what is the best way to use. I will be taking some of these sessions there, and I hope you will be enjoying that session. And it will be a very, very learning, inspiring session for all of you. Looking forward to see you in the class. Thank you very much.
Thank you so much, Dr. Kakar. So, does the participant have any queries related to the field? If they can let us know in the chat. And without further ado, let's also start with the next part of today's webinar. I'd like to invite Dr. Mohit Mazumdar, who will also be the other program mentor for the Cheminformatics program. He has over 12 years of research and industry experience, and he has completed a master's in bioinformatics from Jamia Millia Islamia and has a PhD in computational biology and chemistry from Jawaharlal Nehru University. He is an expert in machine learning in application to biomedical and chemical data. So without further delay, let's start with today's section. So the stage is all yours.
Thank you, Shri Gauri, and thank you so much, Dr. Puneet, for giving us that clarity about how the training program would be and why we should be all excited about it, because it's a time where technology is evolving at a rapid pace, and at the same time we are finding new things, new details, and doing discoveries. So it's a great time to learn this and start applying, because the opportunities are like that only, where the industry is providing the right kind of opportunity if you have the right kind of skills and experience. So thank you again for joining today's session, and I'm really excited here today to meet with you all who are interested in utilizing modern data analysis tools, learning and exploring the 2D, two-dimensional, as well as the three-dimensional space to find the answers to the problems of drug discovery. So we have been discussing in the session that how this training program would be. So as Dr. Puneet said correctly, that it involves the traditional learning about the basics, learning about what all technologies have been used so far in drug discovery, and what are the case studies where this success has happened in this utilizing data-driven drug discovery. So we will be exploring techniques that will involve us to utilize more both two-dimensional and three-dimensional space. So it's really exciting to be able to do that and develop such skill sets as well. So about the entire process, So we will get to the questions also in this session. So in this presentation, what will be covered in my part of the presentation, and we'll find time for to address the questions. So please feel free to put your questions in the chat, and we'll be able to address them in between. So as I was telling you that the process of drug discovery, it demands the selection of disease, as we all know, target hypothesis. So what would be the target and the hypothesis behind the target, and searching for potential compounds that that would be helpful for us to reduce the severity of the disease to any extent, right? So these are some of the very critical components when we think about drug discovery in general. And then there are different stages in there. So lead identification is a stage, preclinical trial is a stage, clinical trial is a stage, and then one could say that pharmacogenomic optimization is also a stage. So what are the different opportunities now and challenges that the industry is facing? Because when there is like opportunities and when there are challenges, you will find also the opportunities there. So one of the very talked about discipline that you might have been hearing or research areas, precision medicine, so personalized medicine. And why this program is a modern data discovery or data drug discovery program is because of that, because we are not only thinking about drug discovery as a traditional way of looking at it, but also a modern way of looking at it. Why? What are the different ways that we can enhance and reduce the time, as well as find meaningful answers without fail getting failed in that process? So precision medicine, drug design is an exciting field, drug repurposing is an exciting field. So in this presentation, we will learn about the modern drug discovery pipeline that we've been discussing about, the cheminformatics and data-driven drug discovery, the training part and the project part, the schedule of the online training and the research. How are we going to do it? What are the ways one could register, and what is the sustainability cost for us? And also the advancements and the use of cutting edge technology. So from machine learning, deep learning, to utilizing other known data driven technologies, all those options are there. It would be based on the progress, how you progress and how you interact with us and learn through this entire process. So a little bit of introduction. So the drug discovery program that we have started, which is the Cheminformatics for biomedical drug discovery, and this is the current version. We started this program in 2020, where we collaborated with the pharma industry, and one of the key associate partner of us to be able to do this is the Faba Academy. So Faba Academy, along with their mentors, we designed and we first partnered, and then we designed topics and curriculums and learning modules that will be more relevant for the students, because it's driven from the industry, experts from the industry, leaders from the industry. So the first course that we did in 2020 included advisors from different companies who have a rich background working with big pharma and biotechnology companies, and now our co-founders and founders are working in this space. And you have already met Dr. Puneet, and you will also find video presentations by Dr. Ajit Kamath, Dr. Uday Saxena. And if we get an opportunity, if you start working on your own project, then we will have a discussion, and we will have them as also help you in this entire learning and research training program that we are just inviting all you to join. So that's about the conceptualization and how all of this has started. It started with an industry aspect, and it focuses on the gaps that are not being filled in the education or other trainings that are available. So a little bit about the modern biomedical drug discovery pipeline and how things have changed. So people who are learning drug discovery, they know about docking, they have heard about simulations, molecular modeling, AlphaFold, and all. But these are tools. So before the tool, I think it's very much important to understand that why we are doing this, what is how we are doing this is something that will come next. But why we are doing this and what all we can do is very important to know. So that's why this omics logic training program is a great opportunity, because you get to know not only from the drug discovery aspects, but from a genetic or a more detailed from a more detailed data that will tell you that why why there is a gene, why there's a mutation, and why what is the effect of that mutation. It will help you to find new targets. So genomics and biomedical data sets, they help us identify, for example, biomarkers. Now there is a company called DNA Nexus that focuses only on this, like genomic and biomedical data sets they use, and they identify biomarkers and they focus on drug discovery. So another important rich area to find information to start your work or to find something new is through the scientific literature. So scientific articles to identify target ID. There are large number of companies, and they just focuses on this, that identifying articles and the conference presentations, and from the value that the academicians are generating, and to find the identify drug targets that are relevant for the industry. So several companies work on that, even in India. Another aspect, so tumor sample data to identify prognostic biomarkers. So this is predictive oncology, huge application in oncology, as for the known reasons, very complex disease and very less recovery. So a lot of opportunities are there in terms of digging the data, finding the right kind of target, and then targeting molecules for that kind of target. Medical literature and symptom drug data for drug repurposing. So we've been talking about drug repurposing also. So the drugs that are been already approved, if they are working for other indications, how do we know that? What is the method? So there are companies like Helix who are focused just on that. Cellular fluorescent imaging data for richer in vitro pharmacology. So there is a company called Recursion that works on that. Protein structural data to identify peptide binders. So there's a company called maintain.ai that focuses on that. So protein structural data and compound binding for small molecules hit generation. Atomwise is a company that works on that. So as I'm telling you that, like, different disciplines, even a micro discipline within a discipline is enough for a company to work on that, finding different types of ways to assist the drug discovery process. So the opportunities are so much in this space. One would need a understanding of how to work with the data, how to analyze and find meaningful way to analyze the data. So we've been discussing about the application of modern biomedical drug discovery, for example, in personalized medicine. So personalized or precision medicine, initiating from clinical and, like, for example, from clinical data, from molecular pharmacological data. So it introduces a new era in healthcare, and it aims to identify and predict the optimal treatment for a patient or for a cohort. So this is also like earlier called evidence-based medicine. So evidence-based medicine integrates information which is what you see in vivo, and which is what you see in the observational studies, which is what you see in clinical trials, up to the meta-analysis for the clinical consideration. So this is to understand the population, also like the average of the population. And there are many companies who are intensively working on this to understand the pharmacological aspects and different aspects that could help them in novel drug discovery, drug delivery systems, and also drug repurposing. So this research will allow us, like, will allow you to utilize both experimental or clinical data. So you have that understanding that how things are working in real world. and then design a project. So many of us come from an academic background, and many of those projects are not translational in aspect. So one of the things that we are trying to do here in at Omics Logic is to work on translational problems. So that's also an opportunity. Now, application like understanding, for example, this is just to elaborate on that understanding that how, for example, transcriptomics. So transcriptomics is to understand the transcriptome, which is the gene expression patterns. And how do we find out things? It's very simple. Like when you compare, so understanding the consequences of an individual transcriptomics variation. A individual means a patient, which differs from any other patient. So understanding an individual's transcriptomics variation is kind of fundamental today to decipher the human biology and disease, because this data is something that you need to look at to understand and like gain all the insights that you might require. So gene expression patterns in association with clinical data can help identify molecular biomarkers for diagnostics and for therapy selections. So new discoveries include a better understanding of these complex diseases at a subcellular level, as well as target identification for novel drug targeting or drug designing or repurposing. So this multi-omics approach is the modern drug discovery that uses, from sequence to structure, from omics insights to understand the cellular interplay, protein complex interaction, understanding the entire environment, which is the system biology approach, and then trying to mimicking that environment, which is through molecular dynamic simulation. So that's after that you have all the computational tools. In between you're doing the discovery, in between you're trying to model and find the right kind of complex, which is also a difficult task, because there are so many small consideration you have to make when you are trying to mimic something within an organism, for example within a host. How the molecules are acting within inside our body, they are not stagnant, they're moving all the time, there is dynamics involved. So how do we able to predict that, and to predict them correctly is a challenge. So, and that's why we are here to as a mentor, and we have developed all these sessions and materials, and utilizing the research which is ongoing, the current research, and our own experience to help guide students achieve and learn these technologies that will be really helpful in the future and also now. So from target discovery, we've been discussing about target discovery. So we discussed about how different types of clinical samples, cell line data, cell model data, patient samples, animal model data can help us in that process of, target identification, then target validation, and then go for drug discovery. So this initial phase is critically important if you're thinking about an entire big project, or if you want to work on a small project also in some of these associated areas. Now, about the training that we are going to start, the Cheminformatics for Biomedical Drug Discovery has all those aspects. So quickly, we will get to an understanding of how the application of machine learning algorithms are there, how the application of different types of approaches are there. We'll start with an overview of that. And then I'll tell you about the sessions, how the sessions are planned, and what we will be covering in the sessions, and how that translates into a independent research, or how that translates into research. So one of the things that has happened in the recent past, and if you guys have any question, feel free to write it in the chat and during this presentation. I might, maybe before I start this section, just take a look at the questions if there are any. So Dara, how can one incorporate cheminformatics, a dry lab process, with animal study, a wet lab? So as I was telling you, that what wet lab studies are doing, are helping, is they're helping in generating data. Any wet lab experiment will generate some data, and once you have that data, it's a dry lab process. So it's all correlated. You just have to know that what kind of data you're generating with dry wet lab experiments. If you know about if it is a cancer related research, and if you are generating some data with that wet lab experiments, you can reach out to us and we can discuss how that data can be further, how your even experiments can be designed based on data. So the approach has changed completely now. Now we look at data first and then we design experiments, and that's how we are cutting the cost. Earlier we were doing virtual screening with millions and millions of compounds. Now it's knowledge driven. We know that, okay, this is the active group, this is how the molecule should look like, these are the different molecules that are good binders. So we go with information and knowledge, and then we go for a specific experiments. So it is what it is helping is that it is helping in reducing the cost so much, like from these high throughput experiments when where you're trying everything against everything. Now you're very focused, and then the chances and the probability of your success is also very high because it's knowledge driven. uh Devpreet, there's a question. So we are posting these jobs on our Omics Logic portal also, so we'll share that job listing and you'll be able to see that in the portal itself. But we are also have a LinkedIn group where I share about these job opportunities all the time, like every day. So you'll find jobs there also. And like you can also search out, look for yourself with these keywords, and you'll find jobs and postdoctoral opportunities, PhD opportunities, even master scholarships and all of that through this sort of training. We have students who have completed it, and they have gone to do higher degrees in very reputed universities through recommendation and through the experience. Yeah, I think our team member, Devpreet, our team members will be able to. Yeah, thank you, Shubham. And please help Devpreet. All right. So now going back to the second phase of this. So I wanted to discuss the application of machine learning algorithms in different aspects. So in most drug discovery projects, what happens is that the protein is chosen as a drug target, right? For the development of therapeutics. So that's how we know things like, so several supervised and unsupervised machine learning approaches have been used for structural prediction at different levels of analysis. So we are first talking about structure prediction. So if you don't have a structure which is your drug target, how do you predict that structure? So that is something that comes first. But if you already have a co-crystallized or structure, predicted structure, or sorry, experimental structure, then obviously there is a high probability that because all the atoms have been mapped with an experimental map, which has been generated through maybe X-ray crystallography or NMR spectroscopy or cryo-EM. So these are experimental methods. And if you have a structure which is derived experimentally, then the probability of all the atoms present at that 3D space is very high. So you get like maybe you get better results. But then again, when you're doing modeling, the structure that we get from modeling is also very detailed. We just have to make sure that it's properly modeled. So some manual maneuver is important in that context. You look for missing residues, because many times what happens in modeling is that in crystallography there are some regions where there is no electron density. So in that case you have to find and build those areas. So how to make sure that those are stereochemically physically like stable, low energy. So all those considerations are important when we are thinking about uh, like predictions. So in, as I was telling you that again, like predictions happen at different levels. So primary sequence of amino acids, then finding the secondary structure from the primary structure analysis from the primary amino acids. So from, you have 20 amino acids and your string of them as a protein sequence, then you want to predict that how they are going to fold. So whether they will be alpha helices, whether, or there will be beta sheets, or there will be coil or loops. That's a prediction. That's how it starts from the secondary structure prediction, then the tertiary structure prediction, that how it is going to fold in a three-dimensional form. So the problem of protein folding. So you're trying to solve that. And then quaternary structures and protein structures, how it is interacting with other three-dimensional structures and how it happens. What are the insights that you can get? So machine learning algorithms are used for secondary structure prediction, solvent accessibility prediction. So what is the solvent? How many areas in the protein where solvent can be accessible? So your solvent could be water also. So to find out through the grooves and all. And then topology generation, contact map generation, which finally integrates this knowledge into building a 3D model of the target. So supervised machine learning methods like SVM and DT algorithms, they are used for rule generation, training data set, for secondary structure prediction. Artificial neural network is used to predict distances between the pair of amino acid residues, which is used to construct the potential of mean forces for generating protein shapes. So these are all published studies that I'm talking about. And the machine learning methods are also used for fold recognition and model evaluation. So then we have molecular or 3D building techniques, or like the algorithms that have been really used from very beginning, like conjugate gradient descent model. So the conjugate descent gradient model is also used in energy minimization. Monte Carlo methods are used in optimization, energy minimization, as well as 3D model building. So these algorithms or methods are like traditional algorithms, and now they are working with the machine learning algorithms. So in model generation, you get to work with like different types of algorithms, and you have heard about AlphaFold from DeepMind, that performed well, and they created models, and they have now a database where they have predicted all the models in association with EMBL. So what are the different limitations and all, we will also learn about that. But like, as you have have a now clear understanding, I guess, of what is the application of machine learning for primary sequence of target proteins. Now the second is a ligand molecule. You could see that what are the like ligand, from how do we go, like we learned about one thing, which is from structure to finding those inhibitors and molecules. Now from the other approach, for example, give you this ligand molecule, the right side of the panel. So a single bioactive ligand can be used as an input to search for a vast set of similar compounds. So you have a known drug, and it's a bioactive drug, and it's working really well. Now you want to find similar compounds in the chemical databases. So machine learning approaches such as kernel discrimination and naive Bayesian classifier methods are used for such virtual screening by analyzing the property of the bioactive part. So similarly, compounds are searched, scored, and screened based on the distance-based similarity, functional group matching with the bioactive input bioactive compound, and then the distance-based approach as well. So this includes like, chemical fingerprinting, functional group like shape, pharmacophore, and all of that. So this is an overview of all the things that are happening, and advancements and integration of the methods that have been used, such as for ligand-based searching. Now we will learn a little bit about the other two ways, which is like de novo design of molecules with desired properties. So this is also a very hot topic, and it's being used by some of the AI-based companies, such as in silico medicine, to generate molecules of desired properties. So computational approaches can be used to generate chemical molecules as well, with desired physiochemical and biological properties. So supervised machine learning algorithms are used for training the model to generate chemically feasible smiley strings. So smiley is a format to generate or to write chemical structures, and also for accessing the biological property of the compound. So there's a There's a deep learning method called reinforcement learning for structural evaluation, which is a deep reinforcement learning based method. And also there are graph theories which are used for all of this. So this was a brief overview of the advancement that one could use. But for those who are starting and those who are beginners, obviously we will not go to the advanced steps just like that. So in this training program, we will cover from a very basic, like we are very big beginner level, where we have started, where I have started understanding what are the basic things and how to understand and relate from this basic understanding to different stages, like from basic to 2D space to 3D space, and then machine learning, deep learning, molecular dynamics, all of that. So it's an exciting journey. It's and very visual, because we will be doing hands-on in throughout the training program, because it's mostly hands-on and very less theoretical in aspect, because theory you can always read and learn from research publications and Google. So here we will start with, as I was telling you, that we will start with very basics, like protein structures are very important because they interact and they communicate, and there is chance that there are targets. So protein interactions to how to do molecular visualization, and molecular visualization is very important for cheminformatics or for drug discovery. So all of you who would be joining the program we'll be learning about different molecular visualization tools and how to utilize them. Then we will go into cheminformatics. First, we learn about the macromolecules, and then we learn about the small molecules, and how to explore the chemical space, like databases, prepare for virtual screening, large databases, how to utilize them for drug target identification or for searching similar ligands. Those are the key things. And then key businesses. So there is always a business component or industry component associated with our training here. So then the focus is to go into those databases, explore those databases, learn about accessing those databases, and curate bioactive molecules and find the right kind of molecules. So that could be specific, for example, kinase inhibitors, or you're looking at very specific type of inhibitors with specific type of biochemical or physiochemical properties. So in this, we will be, again, the hands-on part is there, where we will be learning about the softwares, tools which are freely available for you to utilize and use them along with the mentor or after the mentor session, and practice on your own, because we will be also providing the recorded session and the content and the material and the links in a systematic manner, so that you learn from the basic. And even if you miss some of the sessions, you can go back and you will have it all with you to learn and do it anytime you wish to. But it's advisable to go along with the sessions, so that the desired outcome can be you can get the desired outcome within that time space. Now, exploring the protein data bank. The protein data bank is I think is the cradle for structural biology and for drug discovery, structure-based drug discovery. So here we find all the experimental structures, and you can see that as many as 60,000 structures are available for humans, and the list goes on. So we have a lot of structures now compared to the previously, and you can see the number of structures that are deposited and how they have grown over the time. And different, like for example, SARS-CoV-2, just in a few years, like 3 years, there are more than almost 3,000 structures, which is massive in understanding about a virus as small as SARS-CoV-2 and having so much structural information. So just imagine if you can understand the system biology from that, how many opportunities are there to help. So yeah, I mean, this is the repository where we will be finding the structures and understanding the very basics of structure, how to understand the structure, how to change something in there, how to incorporate mutations, how to understand the role of the mutations. and exploring different types of research projects and structures. So here the participants who will be joining, they'll have the opportunity to explore the three-dimensional space and find their own structure of interest, disease of interest, and work with that structure to further investigate and find out how they are folding, how they are maybe functioning, what are the key residues, where to find the active site, what are the solvent accessibility areas, if there is an allosteric site or a conventional drug discovery will work, what are the different types of drug discovery approaches one could take, whether it is a flex, whether it is what kind of docking are we going to talk about, what kind of simulation. So all those things are possible. So we will elaborate on each of these sessions. For example, target structure prediction. I have already discussed a lot about it, so let's not. So we'll not discuss it further. But these are will be discussed in the context of a case study. So where we will be covering these different topics, and then we will be working in that demonstration section in the session during the session. So different types of modeling. What are the different technologies that are used? What are the different tools that are used? What are the pros and cons of using one before another? And what are the most popular and validated methods that are out there? Then target identification and virtual screening. So virtual screening is again very important in that drug discovery aspect because it's being used. So Prashanjit has a question: which software do I need to install on my Mac? So for molecular visualization, Prasanjit, you can download Chimera or Pymol. Chimera is free and a very powerful tool. Pymol is proprietary, but also for academic, there are free versions which are available. So I think you can start from there and have a molecular visualization tool for sure installed in your software, in your MacBook to start with. So now about virtual screening, as I was telling you that virtual screening, so let's simplify this. So virtual screening aims to computationally search the universe of potentially organic molecules to identify a manageable number of virtual hits, right? So what it is doing is that it is searching for molecules which could be considered as hits. So identifying them in the context of how the initial screening could be done. So one such computational method which we use for virtual screening is docking, right? Molecular docking. So it uses the three-dimensional structure of the target protein and places small molecules into the binding site. So there is, however, a computational complexity which is there in docking. The evaluation of each small molecules require consideration of its low energy three-dimensional conformation. So this is what I was telling you about before, that when we are doing virtual screening, when we are starting from virtual screening, there are different steps. So one is virtual screening, another is then intensive docking, which is where it is computing all the different parameters which are also important, which is not done at the screening stage, because screening, you have a large library. So generating low conformational three-dimensional calculating the energy of that conformation is not possible at that point of time. So we have tools which do that and help us screen from larger library to smaller libraries. But obviously, what is the best approach in bioinformatics for me is to utilize multiple tools and then come up with a table which has the top scorers from each of those tools. So that in a way, within bioinformatics, validates our work. So that will be covered in the fifth session. And then we'll learn about the different steps. And we have some projects that we have already worked on. For example, this is a drug discovery project that company has worked on with Roswell Park Cancer Institute, which we will be using as a case study. And then we have the actual session on the other aspect after virtual screening, the in-depth docking and stages of drug discovery process. So we will learn about the pharmacological aspect and drug discovery aspect and the different conformation. using the top softwares which are freely available, but also the concepts are like the logic is same. The software might vary, but once you know the logic, once you know how to utilize it, what are the different aspects, I think the language remains the same. So it's important to understand the basics and also the parameters that you have to look into when you're looking into these different aspects. So one of such experiments, if you do with us, you will be able to explore on your own, and also we'll be able to help you in case you use different softwares of your own interest. So we in till the sixth session, as you could see, we are learning about the basics, and we are learning from a very beginner aspect to for those who are learning for the first time or who want to learn from the very beginning and basic level to the advanced level. I think first to six sessions are to help them reach at that stage, and then we go into more advanced topics such as data science and AI in healthcare, cheminformatics using R, these such topics. So in this aspect, I would like to talk a little bit about QSAR. So QSAR or QSPR, these are based on the idea that the structure of a molecule might influence its physical, chemical, and biological behavior. So we have been using, by the way, QSAR for last 40 years in the practice of agrochemistry, pharmaceutical chemistry, toxicology, and many other facets of chemistry. So QSR has effectively modeled many physiochemical properties such as toxicity, metabolism, interaction between drugs, carcinogenesis. So initial QSR model such as Hansch and Free Wilson analysis, very popular, I guess, uses that use regression models to relate potency and structural and chemical properties such as chelation, substitution patterns, surface activity, hydrophobicity, solubility, electronic forces, electrostatic forces. So in the context of the overall protocol for constructing QSR model for drug designing has been systematized, and many open source tools and softwares are available for such steps which consist of several modular steps for chemoinformatics and machine learning techniques. So in QSR modeling, you start with molecular encoding, in which the chemical structure of the drug molecule is used to derive the chemical features and the property, and then you go on and work on that. So there is an opportunity for those who want to learn about this in the advanced modules that we have prepared, followed by the case study. So we have a case study which is the discovery of sirotinib kinase inhibitor for lung cancer, and also there's a blockbuster drug that recently got there's a company that was recently got acquired by BMS. BMS is a Bristol Myers. It's a very big pharma company for $4.1 billion. So and it's a inhibitor. It's a kinase inhibitor, and it works on different chemotherapy treatment. So I think, I mean, that's some of those case studies are really helpful for understanding that what are the success stories, and also what are the different ways these companies have utilized to reach at that stage. So, I mean, tools and softwares, here's a list from Autodoc to molecular docking to ab initio modeling. So we will be able to utilize many of these for the understanding, unless there are proprietary, which is like Desmond, Schrodinger, or Charm. So other than that, the ones that are available, we will be able to use for the project work. If you have a hypothesis, if you have a structure that you have started working with, or if you have a disease type that you are working on. So those who would be joining. So we have different modes of joining the program. So one could choose to join between for 1 month or 3 months. So for 3 months, we will be able to work on research proposals. And then we also have the research fellowship program, which actually helps students to complete a research project. So then as I was telling you that this entire drug discovery is focused on the modern approach. So you can work on oncology related projects, you can work on infectious diseases related projects, and you can use multiple types of data types to be able to come to the drug discovery or drug target, or you can use several other approaches that we can work with. So it's up to you, and it's also you will be also learning about these approaches. If you have not if you think that you're new to all of this and you're not ready for it, so that's why the beginner level is there, so that you are ready for all of this. So again, here the focus is AI in molecular drug discovery. We discussed about artificial intelligence playing a role, and also deep learning has a huge role. So we'll discuss all those aspects in this session. And then understanding that from the point of view of how these different technologies. So Google has DeepMind program, has the open AI program, where the Dr. Puneet just discussed about Chat GPT. So incorporating the new technologies, the real really impactful technologies that are came into 2023, 2022. So I don't think there are any other programs which utilizes that, but this is what we've been doing. We are updating it like every month, every week, and so that we can enable those who are joining to get to the latest technologies, learn about the latest advancements, and apply them into their research. So this will include understanding, and this would include pharma and bioinformatics industry integration. This will include like different types of softwares for molecular visualization. This will include advanced cheminformatics, which is to further manipulate the structures. This will include the research projects that I've been discussing about, and this will include integration of omics data in research projects, if that is what your direction is. It will include you to work on pharma projects, which is for specific pharma problems. It will include for you to work on different infectious disease projects that are there, for example, SARS-CoV-2, and learning about different binding properties. We have already published articles on malaria vaccine design and resistance using popular population genetics and drug discovery and molecular dynamics on all of that. So in the end, we have a case study which is on successful as well as the failed drug development, to give you an insight about what worked and what has not worked. This is a recent publication that we were able to complete through this training, and it's an infectious disease genomics research which was recently published in Molecules. So I think that concludes my presentation for today. And we have still the registration and the cost and the scholarships, and really exciting time, because we have a really great scholarship model which is just available for few days, which is I think what Sparsh will be talking about. I'll look into the questions, and I'll meanwhile I'll also invite Sparsh to discuss about the registration, and through those address any questions. I'm checking in the chat.
Thank you so much, Dr. Mohammed.
Yes,
okay, thank you, sir. So let me share my screen. So could you please confirm if you can see my screen?
Yes, we can see it. You can see it.
Perfect, thank you. Hi, my name is Sparsh Dhar, and I'm the marketing manager here at Pine Biotech. So I'll quickly guide you all through the registration process of this particular program named Cheminformatics for Biomedical Drug Discovery. So this is the program page, and the program is going to commence from 27th Jan. And in between, we'll be having one more webinar, which would be related to a hands-on session. So we'll be telling you about that particular webinar in a short while. We'll be sending you invitation about it. So when I'll scroll down, so about the program overview, the curriculum, etc., Dr. Mohit has already explained it in a very comprehensive and extensive way. So I'll directly jump onto the registration process, the resources, costing, and the scholarship part. So here you all can see there are three different tenures in which one could enroll for. So there are some of the resources common in all the three tenures that are online tutorials that you will be having access to the Omics Logic Learn portal, which have more than 32 plus courses on different domains and area of analysis. Then we have more than 16 plus example research projects again on different area of analysis and domain plus case studies. Then you will have access to the project examples, the coding sessions, the session recordings, online mentor support, online technical support, certification of completion, and example cloud pipelines on T-Bioinfo server. So these are the common resources which one would have access to if they'll enroll in any of the tenure. Now, what is the difference between the three tenures? In 45 days you will be getting a full-fledged training. You will be trained related to drug discovery, cheminformatics. You will be taught different software tool databases, etc. In 60 days tenure you have two different opportunities. So the first 45 days would be devoted to your training part, and the next 15 days would be devoted to how you can draft a research proposal, present it to the mentor, so that the mentor can evaluate your research proposal and guide you the changes or any amendment which would make your research proposal more validated, and further you can work on your independent research project also. One. Second is you will have access to one-on-one mentor sessions. So for example, if you are going through any training module and you get stuck somewhere and you want to meet mentor one-on-one, so we can fix an exploratory call of your with the mentor of around 15 to 20 minutes, and you can clarify each and every question. Now, the outcome for 60 days is a poster presentation. It could be in a omics research symposium or any other research symposium. It could be in any international conference also. Now, in 90 days, you again have two different opportunities. First is one-on-one mentor sessions, obviously. And second is the first 45 days would be devoted to your training part, and the next 45 days would be devoted to how you can develop your independent research project. And that particular project would be further publishable in a good peer-reviewed journal. So the outcome comes to be a research publication and also a poster presentation. So this was the difference between the three tenures. So now you can see here on my screen, the pricing is in USD, and you can definitely change the pricing according to your location. So here there is a drop down menu, and in this drop down menu there are different currencies mentioned according to different locations. Now if I'll click on USD and I'll scroll down, I can see the pricing in USD. So for 45 days it's $150, for 60 days it's $300, and for 90 days it's $450. And now till January 15 we are providing each and every interested participant with a 50% scholarship, and one of our representative will publish or will tell you the steps of registering, and will also tell you about the coupon code, which is CHEM50, CHEM. CHEM is in capitals and 50. I'll just tell you how you can proceed further with the payment checkout process also. Now, for those who are in India, so the pricing is in INR, and when you scroll down, you can see that for 45 days, the pricing is 7,500. Then for 60 days, it's 10,000, and for 90 days, it's 15,000. And likewise, you can change the currency depending upon your location and see the pricing or the fee for the program accordingly. Now, moving further, telling you about the payment procedure. So for example, if I'm interested in 45 days tenure, I'll click on the Buy Now button. After clicking on Buy Now button, I'll be redirected to the Omics Logic checkout page. So I'll just quickly type here the coupon code, that is CHEM50. Click on Apply, and here you can see that the coupon is applied successfully message will pop out. You will need to
fill out your user details like your name, your email, your phone number, your country, occupation, organization, etc. Click on this checkbox and select the payment mode if you want to pay it via Stripe, PayPal, etc. Now, those who are unable to pay via PayPal or Stripe, we have alternative payment methods also. And for that, you can uh, write us at marketing@omixlogic.com, or you can ping us at the official number, that is +919876134120, and we'll guide you with the alternative payment methods also. So this was about the registration process, the scholarship, and the fee. So if anyone is having any doubt related to the registration process, etc., please feel free to unmute yourself and ask, or please feel free to write in the chat box, and I'll be there to assess and guide you. Thank you. So Sri Gauri has already shared my details. So the marketing, so email ID is marketing@omixlogic.com, and the contact is 9876134120. This contact number is active on WhatsApp also. So those who want to connect with me via WhatsApp, or have one-on-one conversation via WhatsApp call, that is also applicable. The coupon code again is CHEM50. Expiration of the coupon code is on January 15th. So please hurry up and avail the opportunity. If anyone is having any doubts, so please feel free to write in the chat box. Over to you, Sri Gauri. Thank you.
Thank you so much, Sparsh. So we'll pause for a couple of seconds. If the participants have any queries, you can either unmute yourself and ask, or put your query in the chat. Otherwise, we'll be ending with today's webinar. All right, then I guess Prasenjit is asking, what software do we need for hands-on training?
Yeah, so to address that, Prasenjit, you can start with the molecular visualization software, and then we will be sharing throughout the program the tutorials. You would need to install the softwares that would be needed, but many of the softwares are available online. So the web servers will take care of that. So you'll not have to install a lot of softwares, but those softwares which requires high computational power, those will be required to install, and you can start with the molecular visualization softwares, and then we will share with you as you join the program. Once you join the program, we will share with you the detailed tutorials.
Thank you so much, sir. Sparsh, there's a query for you. That is from Tanmoy. Are there any discounts for BSc level students?
So yes, we are giving right now 50% discount to everyone for every level of education.
Thank you so much, Sparsh. And Wahid is asking, can you recommend few free software for molecular talking?
Yeah, Wahid, you can write an email to me or our team members, or connect with our team members, and we'll share with you over an email the free softwares that you can start exploring with.
Thank you, sir. And we have another query from Mohammed Noor. Does your platform provide online software or any interactive practice?
We will be using open source softwares, and the platform has a structural biology section where that could be utilized for molecular screening.
Thank you, sir. So if the participants have no further queries, we'll be ending today's meeting. So have a great day, and we look forward to seeing you at the program. Thank you, everyone.
Thank you.
Thank you, everyone.